Multicellular tissue is complex. This protocol reconstitutes cell-cell contact in a simplified manner. One can use this technique to study the direct relationships between cell contact pattern and cellular response. This protocol uses chemically functional polystyrene beads to reconstitute cell contact patterns. The beads can be coupled to any proteins of interest to test their effects on cellular response. We have used this method in C. elegans and mouse embryos. Therefore, this protocol can be applied to the studies of wild organisms.
As embryos are fragile after hypochlorite treatment, significant practice is required before one can successfully isolate blastomere. Visual demonstration will provide a greater understanding of how one should handle capillary during blastomere isolation methods.
To begin, weigh 10 milligrams of carboxylate modified polystyrene beads in a 1.5 milliliter microcentrifuge tube. Add 1 milliliter of MES buffer into the tube to wash. Vortex the tube to mix the beads. Spin the tube for 60 seconds at 2000 times G via a benchtop centrifuge. Discard the supernatant by carefully pipetting out the buffer. Wash the beads again with 1 milliliter of MES buffer. After the wash, add 1 milliliter of MES buffer containing 10 milligrams of EDAC into the tube to activate the surface carboxyl groups. Vortex the tube to mix the beads. Rotate and incubate the tube for 15 minutes at room temperature. Spin down the beads for 60 seconds at 2000 times G. Discard this.